Okay, I'm going to start with something a little bit different and potentially, as you look at this slide, you're like maybe a little bit intimidating as well. I had a graduate student in microbiology. He showed up to my office Monday afternoon. We had just lectured about this stuff. We just talked about the lac operon and all sorts of potentials. What if glucose, or what if lactose, or what if both are present? Different potentials for the bacterium to maybe utilize this stuff. And we talked a little bit about some of these details. And he rolled into my office and told me he built this thing, which made me wonder, what the hell, right? That's pretty impressive. So he shared this with me. I, in turn, did not get a chance to get it downloaded and posted to Moodle before class started this morning. So I opened it up, but I will tell you, I will have this file loaded onto Moodle a little bit later today, if you are interested. All right. I feel like potentially there are some customers in here that might be a little curious about some of the genetics and maybe want to mess around with a little of this just to see what happens. What if I did this, or what if I did that? And this, he technically had a couple of different ones, but I was like, the lactose one is what we talked about a little bit in more detail. And it's definitely a little bit easier to understand what's up. All right. There are a couple of features in here that maybe not. We've got some wild types and then some potential mutants, which you could manipulate all of these things and see what happens. All right. So initially, we've got our segment of DNA. We've got all of the respective regions, these genes in here. We could do lots of things. All right. And once you get access to this, in his message, he did say that it likely needs to be on a laptop or desktop, not just whatever the open Excel is. I don't know where you'd be getting that on your phone. That probably doesn't work. All right. You have to enable editing. You have to enable macros, and then everything should work. All right. Because all of these things are all embedded in there, which some of that stuff is completely boggled my mind as he started showing me stuff. So you can manipulate things. We could label stuff, maybe. So we could see regulatory genes, regulatory region, the genetic region, the business end of making those enzymes to deal with the lactose in the first place. And in blue, this whole chunk of material is the lac operon. And you can see multiple genes all together, like we said, assembly line sort of an idea. All right. So we could come up here to the top. We've got cyclic AMP going on up here, and hopefully you recall, maybe, this is something that's necessary to get this to initiate in the first place. As glucose levels start to get depleted, we convert some of that ATP into cyclic AMP, and that in turn is one of the triggers to get things initiated. So we could mess around and add different things. Polymerase, we could just go through and add all of these things. Well, I'll leave glucose off. Add this stuff. Now we've got lactose. We'll see if this works. I click go. There are lots of details that happen in there, but as the lactose links up to that repressor, causes it to drop off. And we start going through with our transcription and translation, making our respective products and ultimately dealing with the lactose. If we slap, so we can see over here in green, this is the stuff we turned on. If we get rid of our lactose and slap glucose back into the mix, nothing's happening, right? Our polymerase can bind here, but the repressor is still present, so we don't move forward, all right? We could slap some lactose back into the mix, right? But we still have glucose, right? And in turn, we're going to go through this process because the lactose was present, but our products that we're seeing over here, not the same as, let me get rid of lactose or glucose again, sorry. As if glucose is absent, right? Now we got much bigger arrows and we're actually really utilizing this stuff. I think this is pretty cool, right? There are also some potentials we talked about at the tail end of the last chapter with some of the transposition, those transposons. What happens if things get manipulated around? And you can do that here as well. You can drag and drop and move these things around and see, well, what happens if they're in the wrong orientation or not the wrong orientation, the wrong place, right? Pretty cool. So you have the opportunity, you don't have it yet, but you will have the opportunity to check this out if you are interested, right? I do think it's kind of interesting. And if you feel like, a little bit of genetics is something that you want to go down that avenue for a minute, maybe this might be something that is of interest. All right. Stop that. Back to business. Okay, and like I said, a little later today, I'll try to get all of that posted on there. All right, and if I remember correctly, I think he said he had built that right during pandemic and trying to deal with, how do you talk about these things? Maybe visuals might be good. And I'm just like, I don't understand how you built that in the first place, but that's okay. You're going to share it with me. We're good to go. All right. So with molecular cloning, we left off, I think we talked a little bit about this, but we left off right around here, where we think about the potential to take a gene of interest from some organism and pop it into somebody else. And with that, we've got the potentials, maybe we'll see here in a second with a gene library, where we've got multiple ones incorporated into their own respective plasmids, popped into some bacteria, and then those bacteria, as they're grown up, we can see the expression of each of those respective genes of interest and see what sorts of stuff is happening, all right? It also opens up the potentials for us to manipulate some of these things. What happens if we do a little bit of alteration? Just like that lac operon that we looked at, and I said, you've got the potentials to do some manipulating with it if you are interested. What happens if we do some of that stuff? What does it do to the function of that specific product, which typically we think about being some type of protein in the first place, all right? So as this is occurring, we are getting some sort of recombinant, some sort of genetically modified organism, bacterium in this case, that we've got opportunities to visualize and see, did it actually insert? Did it insert properly based off of some pigments of the colonies on those plates? It did or it didn't, and you can move forward with that stuff. Cultivating it. You also can prepare some, maybe you want to store these away for forever if you wanted. You could make some freezer stocks of this as you grow these bacteria up, and then freeze them away in the ultra freezers, the -80 Celsius freezers, where they could basically be there at the ready whenever you feel like you want to go back and reevaluate stuff. All right. So that opens up all sorts of potentials, right? And this is the gene library. So we've got, in this case, just a couple fragments of DNA, and we've color coded them nicely, right? 3 different chunks of DNA. We could imagine each of these different genes, and they go into their own separate plasmids, which in turn go into their own separate bacteria, right? And these bacteria become transformed. The transformation remember, is the uptake of DNA. And in this case, the uptake of these plasmids, right? So the plasmids get popped into these bacteria, and then, just like you guys have done in the lab, we cultivate them, we grow them up, all right? Getting colonies of these bacteria and subculturing, or, you know, taking that colony and streaking it out for isolation, growing up mass quantities of each of these individual red and orange and green genes of interest in separate plates or separate flasks. And you've got mass quantities of that, but now you could actually really do some science with it and see what do these things code for, right? And although this is the, you know, making a library, looking at maybe trying to get all of the genome of some organism, this looks like maybe a nematode or roundworm. I'm pretty sure it tells me somewhere in there what it was. Maybe we want all of these details, right? And harboring that in a bunch of bacterial transformants, we've got all of the genome of that specific organism of interest that we could in turn start doing further investigations with, right? So transduction is using a virus to infect or to insert the specific DNA. It's a similar idea, but instead of those plasmids, now it's going to be this virus that serves as the vector to insert whatever it is we want, all right? So we've got our DNA from whatever it is that we're hoping to study. We've got the viral DNA, all right? We digest both of these with the same restriction enzymes, chopping them up into bits, and in turn, we build our recombinant phage, and it ends up packaging up some of this material, all right? And as we've color-coded, same idea as the previous slide with our respective 3 colors. In this instance, we can see them based off of different pigments, and fittingly, the same that we see right here, right? And then we've got these ideas, all right? The bacterial lawn, all right, has these specific phage that have generated this stuff. And in turn, if we were to go in here, there are virus that are harboring this because they're going to be making it and packaging it up, all right? So similar idea. When we think about eukaryotic stuff, and we've already touched on this the littlest bit, and if this is our little nematode, it is a eukaryote. So you can't just take those genes straight out of the nucleus of this cell and expect that our bacteria can do anything with it. So we got to take a couple extra steps, all right? So we get the messenger RNA that's been processed. We've got rid of all of the introns, and we expose that to the enzyme reverse transcriptase that reads the single-strand piece of messenger RNA and builds double-stranded DNA. And we call that cDNA, complementary DNA, right? It's taken all of the excess garbage, if you will, out of that sequence of eukaryotic DNA, the introns, and given you the DNA that the bacteria can recognize and say, oh, I know what this is, right? I'll do something with it. I'll build these respective proteins, right? So it's similar to what we showed before with our transformants, but it's an extra step to get there, all right? That complementary DNA then is able to be incorporated into the plasmids, popped into the bacterial vectors, and in turn, growing up the respective pieces. Insulin this is how this mechanism truly works. When we think about E. coli or somebody else, but E. coli is a great one for a lot of genetic stuff. The gene with reverse transcriptase giving us the business information of it, and it in turn is grown up in mass quantities. So there's some cells, some bacteria that are normally or naturally competent. So their potential to pull in DNA is more likely. And we might think of some of these, maybe some environmental organisms that are, breaking things down in their habitat. Maybe they're more likely at pulling things like nucleic acids in, right? There are other bacteria that we may prefer to work with because of maybe easier cultivation, maybe growth requirements are more suitable to a lab situation, whatever it might be. But those cells may not be as competent or capable of pulling in the nucleic acid or the naked DNA, plasmids, all right? So we have some ways of making cells that aren't competent, turning them into competent cells, all right? So electroporation is a technique that basically causes some holes, some porosity within these cells, these little bolts of electricity causes some holes to open up, and it allows for the DNA to get brought in to those cells, all right? So that's the idea with that, all right? In this instance, we might be looking at putting it into a eukaryote, maybe into a yeast cell, all right? Instead of a bacterium. Oftentimes, some of the bacteria that aren't competent, we can grow them in the presence of some calcium salts that does, not the same idea, but similar sort of an idea, causing some porosity in those cells and allowing them to become more competent, all right? Just kind of depends. This, I see these images and I think, man, that's awesome. Crazy stuff. You would be using, as we see here, a microscope to really be dealing with this in the first place, right? Here we've got a cell, eukaryotic cell. So we've got our eukaryotic cell. We can see cell membrane, the nucleus of this cell, and this is a micro injection needle, right? Very, very small, right? And in turn, you really need to use the microscope to be able to visualize what you're doing in the first place. But this micro injection, you're able to take that, piercing into the cell and inoculating that DNA of interest directly in there, right? So pretty impressive to actually see that piercing in there, right? There are some, we work with all sorts of different things to manipulate or move stuff around. Pipettes move liquid, right? Like a straw, and there are some disposable or some glass ones. There are some micro pipettes, typically plastic little tips. And then there are some other ones, Pasteur pipettes, glass tubes that are very stretched out thin, almost needle-like, allowing you to dispense little drops of material, all right? And with these micro injection needles, a lot of the time they take one of those Pasteur pipettes, little glass tube, and they heat that up and they pull it really long. They pull it out so that it becomes almost like a very super thin, nearly microscopic needle, all right? That they can in turn poke in there and do their manipulating with. That is pretty stinking cool, I think, all right? So then we can take it a step further. Why not just use a gun? Shoot that crap right in there, right? Wouldn't that be better? So this gene gun basically does that into maybe a plant cell, as we've got here, to pierce through or penetrate through the cell wall of that plant and blast some DNA in there, different DNA, to make some recombinant type of plant, all right? So in this case, they've got different heavy metal particles that are coated with the DNA of interest, and this gene gun shoots it right into that cell. All right, that's kind of the idea. Interesting, all right? And it's using a little bit of helium to blast it in there. We're not using actual bullets, all right? The bullets are the heavy metal particles with the DNA in there, right? So kind of crazy, all right? Another way that we think about manipulating plants is with a bacterium. And in this instance, this bacteria, before we think about it doing the manipulating stuff, this bacteria by itself is bad news, all right? You technically wouldn't like it infecting any of your crops or, you know, trees or whatever it might be, because it causes some serious issues, all right? But knowing that, we can get rid of the issues and just leave behind its ability to get in. And that's really what we'd like, all right? So we can infect, but now we're not causing disease, we're just getting in. But when we get in there, we're bringing in whatever gene of interest that we want into this plant, all right? Kind of crazy. Similar like the gene gun, bringing in different pieces, all right? And it needs to get through that cell wall of the bacteria, all right? So Argobacterium tumefications has a plasmid in there, this chiplasmid that helps us to be able to manipulate it, but it's the bacteria itself that's using it to cause the infection, right? But pulling out the disease stuff and popping in our whatever it is we wanted opens up the potential for all sorts of transgenic plants, manipulating things, putting genes that they never had into them. Whether that's good or not, we will see, right? I think lots of this stuff truly lends to some benefits. And we touched a little bit on some of those potential drawbacks. Just really kind of depends on what that gene was coming out of in the first place, as far as, you know, are there allergic reactions or something like that, right? So the idea with this bacterium, before we've done any sort of manipulating, right? Out in the environment, this bacteria would infect a plant. It gets into that plant, penetrating through the cell walls and causes infection. It causes what we call crown gall. It's big tumifications, big tumors in that plant. And it ultimately causes the demise of that plant, as it basically is like giving it cancer, all right? Not a good situation, right? But we get rid of those potentials, all right? We're not going to cause disease. We're just going to get in, all right? So natural stuff, technically bad news, but once we've manipulated it and using it in a lab situation, we use it to our benefit, all right? So this gives us opportunities for all sorts of different types of transgenic plants, all right? And here we've got our bacteria with the respective stuff in its plasmid in the cell. And in turn, it's able to infect these plant cells as these, you know, like this plant protoplast, little precursors, just individual cells, not a full-grown plant, are cultivated in the presence of these bacteria, and that allows for them to infect, getting in and giving them whatever they want. One thing that I think is pretty cool with some transgenic plants is a type of rice that they call golden rice, right? We're not talking Spanish rice, all right, that looks nice and golden, all right? But that's kind of what I imagine, all right? Rice, lots of starches, lots of you know, glucose tied up in there. But as far as vitamins go, it's not full of a lot of stuff. All right. And there are some places where rice is a staple, and there are some of these places where vitamin deficiency is a big deal. Vitamin A specifically, all right? And vitamin A is essential for vision, all right? We take that vitamin, manipulating it, splitting it in half, all right? And we get, or no, it's the beta carotene that we split in half and we get vitamin A out of it. So we need this stuff for vision. But if we've got vitamin deficiency, we're not getting adequate amounts of vitamin A. And in turn, blindness as a result of vitamin deficiency is a huge issue in some of these areas where we see, you know, populations being pretty malnourished, right? They're not starving to death because they potentially are consuming rice, but they're not getting all the vitamins they really need, all right? So by manipulating this plant and giving it some beta carotene, the golden rice, when you consume that rice, you're able to break the beta carotene down. Now it's like you ate a carrot, all right? You break it down and now you're getting vitamin A just from a bowl of rice, right? And there were two different organisms that they popped beta carotene, the genes for the production of beta carotene. They popped it out of them, placed it into this plasmid, used argobacterium to infect, and now these rice plants are producing the grains, the rice, that are golden. And we can take those kernels of rice, plant those, and grow more rice. And everyone from that point on is genetically modified and gives us those opportunities to produce the vitamin right within it. That's pretty cool stuff, all right? They've taken some other things, thinking like other like drug delivery, different types of maybe vaccines or other things to areas that middle of nowhere, wherever, right? Trucking it for miles to get there, maybe where this substance could have needed refrigeration or something like that. That's a problem. But if you can incorporate it into some sort of food stuff, that's a different story, right? And that increases the stability of whatever molecule you're trying to put in there or drug, and you get it to a population that you may have had super difficulty getting it stable there in the first place. And a lot of it, we think of transgenic plants opening up opportunities to deliver things that we maybe couldn't have before. I think that's pretty cool. So here we can see, well, there's all sorts of different potentials when we're thinking about the DNA and trying to understand what's up. We talked about polymerase chain reactions and being able to amplify all of this material, but we also would like to know some other details about it. Maybe the sequence of that genetic material, and maybe we would also like to utilize it as a detective type of device to see, are we looking at maybe a potential pathogen, as we're looking at in this instance, where we've got specific probes of DNA, a segment of DNA that should, if we've got the pathogen, should link up onto a strand of DNA, as we've done the first hint of PCR reaction, heating up that DNA and causing it to unzip, all right? And if that unzipped DNA is exposed to these probes, little pieces of complementary DNA that are like signatures of this one pathogen, or maybe a handful of different pathogen signatures, and we expose it to this. Each one of them potentially harboring a different colored fluorescent tag, or maybe we're only looking at one pathogen and it's all the same stuff, right? And if it lights up, you know you're detecting it. And if it doesn't light up with that specimen you've collected, well, then you would say at least this culprit isn't present, all right? But typically you'd have multiple probes happening simultaneously, so you don't have to do hundreds of reactions. You could just do this one, right? So the molecular beacon, some sort of tag, fluorescent tag, or some other sort of tag that gives us either a color or the emission of light, something like that, that we could detect, that shows if they've bound, that we've got whatever it is we were looking for, right? That's the idea in that case with the sequencing, looking for specific suspects, all right? So this image, although you could think some of that stuff fitting with it as well, when we want to look at the DNA, we typically aren't thinking about looking at the entire genome all at the same time. That's way too much stuff, all right? But we might like to look at that genome and question, how many fragments do we get if I expose it to this restriction endonuclease or a different one, and potentially expose them to a handful of them and run each of those digests out on a gel and see the fragments, the banding patterns. And we can see that real image down below with our samples in the middle of this stuff. And on either side flanking it would be some controls, some ladders that are known base pair sizes. They run out in there so that you can look at this and extrapolate roughly the size of these fragments based off of your molecular weight ladders flanking it, all right? That's the idea. So the business of what we really want to look at is in the middle, all right? And we can see there's variation with this, different types of banding patterns, and in turn, dependent upon what we're looking at here, we could think about maybe one type of enzyme and a handful of different specimens, patients, whatever it might be, all right? It could be a paternity test, and you've got 3 or 4 different possible fathers. They've donated the DNA. We chop it up with this enzyme. We take the child and the mother, chop their DNA up with the same thing and run them out on the gel and see what happens, all right? You would expect to see banding patterns in the true father. It's 50-50, right? As far as genes that are contributed, half of these things we should be seeing coming from the mom in the kid, and half of them should be coming from the dad in that kid. And as you move through there, that's when we start matching some stuff up. There are numerous lanes in here where we're not going to be matching. You're not, right? There are definitely, we've chopped them with the same things, right? There might be a band or two that we could see matching or super close to matching, but not enough to truly say, this is it. It's when you go through there and you're like, oh, they all fit like this. That's more of the idea, all right? So it depends really on what we're looking at here as far as how we're cutting and the samples we're using. That really matters not, all right? Agarose is a type of gel that is like our nutrient agar, right? But the nutrient agar is pretty opaque. It's got a lot of nutrients in there. It's got a lot of stuff that the bacteria needs. And agarose, we're not trying to grow stuff on this. Agarose is just a gel, right? Something that's going to polymerize and get solid, but it's not full of nutrients. It's not exactly see-through, but pretty close, right? Pretty clear. Once it polymerizes, catalyzes, you can kind of see through it. It's a little bit, it's got a little bit of a color to it, but it's pretty well see-through, all right? As we cast these gels, we heat the agarose up, we pour it into a tray, and we insert these little combs. You've maybe seen a little of this. I think they do some DNA extraction in biology. Is that true? Anybody from biology? Did you do any gel stuff? Everybody in here took 2227. Is that true? How many in here took 2227? None of you, two of you, three of you. Okay, now they're up. Now the hands are coming up. You're like, what? All right, huh? The what is 2227 anatomy? Oh, yeah. So okay, now you're like, oh, anatomy. Okay, now I'm getting more of it. 1101 biology. Anybody do that? Okay, you did 2227 and biology. Yes, you both did. Okay. Okay. Did you do gels in biology? That's all I was asking. Okay. Oh, and here's some chats. What am I getting in here? They're like, what? Yes, they have. Okay, that's what I want. Gels in both? Heck yeah. Okay. Thank you. There it all is. In both of those classes. I like that. All right. So truly kind of depends really on what you're thinking about. All right. You could extract your own DNA. You could expose it to some enzymes. You could run your own stuff out. That's kind of cool too. All right. So the little comb, as the agarose is polymerizing, it leaves wells once you pop it out of there that we can pipette in our samples. There are some dyes and some loading buffer that helps the DNA fit and sink into those wells. And then we expose it to some current and it starts moving through. All right. So the smaller fragments are going to move faster and farther down in the gel than the bigger fragments. Even though this looks solid, there's little pores in there. It's like kind of like a strainer. It allows the water to go through, but not your macaroni noodles or your spaghetti noodles, right? Keeps that stuff in the strainer, right? So the DNA, little bits, small fragments, move pretty quickly through that gel toward the bottom. And bigger chunks, bigger strands of DNA that this enzyme cut, and who knows, maybe, looking here, we've got a boatload of fragments. This is the ladder, right? A boatload of fragments, all individual little pieces, but looking at samples, 2 and small and kind of mid-sized. And this other sample, we got another small one. And then we've got a bigger and a gigantic one up above because it hardly moved at all. It got into that gel and pretty much got stuck right in there and couldn't move any farther. So based off of the ladder, this could tell us, and it's super hard to see here, but it looks like 2000 or so is right around here. Who knows exactly what it is. I can barely see all of them. I'm like, what? That's the idea, giving you the potential to extrapolate sizes based off of that and variation, right? So this, we're looking at some differences with a normal or wild type DNA and then the mutated bit that we can see some potentials that have happened in here. So the RIFLIP is restriction fragment length polymorphism. It's just the bands you get with this one type of enzyme, all right? And you would expect to potentially see a couple or handfuls of bands with this enzyme, and you use a different enzyme and you'd expect to see a different type of banding pattern because they're cutting at different cuts, all right? So with that, we can see using the same thing, same enzyme, right? We've got our normal, our wild type, and this one that's got some sort of mutation, right? And in this case, each of these arrows are showing where that enzyme would come in and make a cut. And the mutation made it so that this bit right here doesn't get cut. And so you would see variation with that. On this other image, the mutated DNA looks like we've got more bands instead of less, right? But similar idea, you'd have some sort of deviation. And maybe we've talked about all sorts of different types of mutagenic things. Maybe you expose this and then you chop it up and run it out and see what's, yes, we've caused problems, but what problems, where were they, right? What genes potentially could have been targeted in that process, and doing this analysis lends some light on that subject. So there are other potentials when we start thinking about more research with it. So a southern blot is going to allow us to get some details about the specific DNA. So the fragments, the DNA gets cut with those enzymes, they get ran out in the gel. So the first bit is what we've just described, and we run it through an agarose gel, all right? After that happens is this middle bit, all right? And we want to transfer from that gel, which is right here where the blue arrows are at. We want to transfer from the gel onto this black line right here, which is a type of membrane, all right? And we use the current to get the DNA to move through the gel. We flip it around the other way and use current to go from the gel onto the membrane, right? Pushing it the opposite direction. So now we've put this onto a membrane, and we are able to in turn use that and probe it with something that's basically going to allow these bands to develop as we see it on here, all right? So it's soaked with the DNA probe that's tagged with those beacons. That's when we flip it back to this, something like this, all right? That now we've got individual fragments of DNA, and we've got our probes that we're basically soaking this membrane in. And if that fragment has complementary regions for this probe to link up to, my southern blot's going to show up as a dark band on the membrane once we expose it to some other reagents and take an image of it, right? Giving us some specific details, right? There are lots of different ideas when we think about this. Southern blots looking at DNA, Western blots look at proteins, right? And try to see what sorts of proteins are in this solution or whatever it might be. And Northern blots look at RNA, all right? That's basically what we think about with this, but we're focusing in on DNA, Southerns, all right? Okay, this is something that it really kind of boggles my mind. Microarrays are big slides that have all sorts of potentials for us to evaluate samples, to evaluate DNA, really, and see what's happening, all right? You can purchase microarrays that have specific genomes laid down on it. So all of the DNA, and this would be wild type sorts of situations. And then we can do some evaluations as we put sample into this microarray. It's basically things that will match up or not, all right? And potential for some flags to show us things that are turned on or turned off. So upregulated or downregulated, maybe either circumstance could be good or could be a problem. Just depends on what we're really looking at, all right? So we've got in this image that looks very weird, right? This is this slide, this microarray is being read by some lasers, and based off of the specific fluorescent tags, we get different things lighting up. And each bit as we move through here, and you can't see any of this, it matters not, but it's a specific gene as we're moving down here. And this would be one organism, a human or whatever it might be, all right? And you can see stuff that's green or stuff that's red, and expressed in this case, the green ones in healthy cells, the red ones in cancerous cells, and in yellow over here, which we've got some going on right there, are expressed in both, right? Giving us some details about what we're really looking at. And we've got two different flasks or petri dishes up here, cancerous and healthy cells. you can kind of see contact inhibition's been thrown out the window as we got a big mound of cells here. And they're all just touching and that's it with these healthy cells, right? So isolating some RNA, reverse transcriptase, and in turn applying this to this microarray and seeing what's turned on or off, right? Giving us some details about some of those specifics at that level. So here we're looking at proteins. So we could see all of the details in this. What are those genes actually coding for? What is this stuff really saying to build, right? So taking it a step further, instead of an agarose gel, now we're looking at PAGE gels. Polyacrylamide gel electrophoresis is what PAGE stands for. And there's really two types, denaturing and native. And most of the time we do denaturing, which is the SDS PAGE. SDS is sodium dodecyl sulfate, basically like a detergent really, that gets added to the solution, and it causes, there's some other stuff added as well, but it causes the proteins to get denatured. So when the protein's in its native state, when it's normally just minding its own business, it's all twisted and folded up on itself, and it's active, if you will. It's able to do whatever protein stuff that protein was going to do, right? If it's an enzyme, the active site's ready and able to do whatever it is, right? But if you denature it, the way it's folded and twisted, now that's gone away and it's no longer functional anymore, all right? And that gives us the ability to see some details about it, all right? So here we've got our protein, big green strand, all balled up on itself. And we denature it by adding it, you know, in this SDS, causes it to smooth out, if you will, all right? And we see this little band instead of the glob of protein. As that goes into this agarose gel, the polyacrylamide gel, now it's vertical instead of horizontal, all right? It goes in there and then the proteins migrate based off of charge, negative up at the top and positive down below. Proteins typically negatively charged, although there's variation. The majority of them are negatively charged, so they want to move toward the positive end of this electric. They want to go toward the bottom. In this case, it's very similar like the agarose, where we can see smaller fragments or smaller proteins moving farther down and big guys getting stuck up in the top, and they can't hardly get anywhere but just the littlest fraction into this gel and they stop moving. All right, so we separate based off of size, but we use charge to really pull them through there. All right, and we can see some variation as we move through, right? Lots of individual bands, all right? We also have opportunities with Western blotting to transfer this to a membrane and then probing it with antibodies that would be specific for individual proteins. And you might see some of these on the gel, they're boatloads, and on the blot, there's maybe only a couple, because only a couple antibodies were able to be produced with these respective proteins. That's the idea. All right, I'm going to stop with this. We will wrap up the last bit of this on Friday. Farewell.